Good morning to SSE students. Today we're going to start the revision for Unit 4, Science and Economy, Chapter 1, Biotechnology and Immunology. But first, please find the correction of the latest homework that you had in this slide. First, we're going to start with a small introduction. Chromosomes are located inside the nucleus of the human cell. Chromosomes are also the carriers of genetic information, and they are made up of chromatin, which is built from DNA and histone proteins, a certain type of protein. Gene is a DNA fragment that codes for a certain characteristic, for example, the synthesis of a certain polypeptide, protein, example, insulin, hormone, amylase enzyme, growth hormone, etc. Activity 1. Principles of Biotechnology. To follow up with me, please open your books to page 126. We're going to start first with the term transgenesis, or what we call recombinant DNA technology or genetic engineering. What is the definition of transgenesis? Transgenesis is the process of introducing an exogenous gene from outside the body, called a transgene, into a living organism so that the organism will exhibit a new property and transmit that property to its offspring. E. coli bacterium is most commonly used in transgenesis processes. The DNA that integrated the exogenous gene in it is called recombinant DNA. The individual that integrated the exogenous gene in his genome, which means his genetic material, is called transgenic individual. What are the steps of recombinant DNA technology applied on bacteria? Step number 1, extraction of DNA from the cell. Number 2, cut the DNA using restriction enzyme in a way that the gene of interest is obtained. Restriction enzyme is an enzyme that cuts in specific locations in the DNA. Step number 3, extraction of the plasmid, which is an extracircular DNA found in the bacterium. Step number 4, cleavage or cutting the plasmid by the same restriction enzyme previously used. Step number 5, ligation or the fusion of the gene of interest in the cleaved plasmid using ligase enzyme such that a recombinant plasmid is obtained. Step number 3, the recombinant plasmid is inserted back into the bacterium. This process is called transformation. Step number 4, gene cloning by allowing the transgenic or recombinant bacterium to multiply. Step number 8, and the final step, is purification of the desired gene or protein after its synthesis. Now, why do we use specifically the Escherichia coli or E. coli bacterium in the transgenesis? Reason number 1, its genetics are the best known of any organism. Reason number 2, it is economically cheap. Reason number 3, it grows fast and can be manipulated genetically. Reason number 4, it accumulates many different proteins. What are the applications that recombinant DNA technology can be used in? 1, we can produce therapeutic drugs. 2, we can produce vaccines. And 3, we can produce monoclonal antibodies. We're going to see in the following activities the applications that are listed here in more details. Kindly make sure to copy the content of this video neatly on your copybooks, and if you have any questions, please let me know. We'll continue this chapter in the following videos.